Hello everyone, welcome you all in our YouTube channel. In today's video lecture, we will discuss about next part of this third chapter, plant tissue culture. In in that types of culture, there are two types. First one, that is depending on the part used for depending on the medium used for culture, and second one, that is depending on the part used for culture. Okay, there are two types according to medium and part. Okay, plant part. In first lecture, in previous lecture, आपण काय बघितलं? Depending on the medium used for culture. Okay, in that continuous lecture culture, subcontinuous culture. Okay, closed type, open type, in that turbidostat, chemostat. These are the depending on the medium used for culture, continuous culture and subcontinuous culture. Okay, batch culture. These are the three types comes under suspension culture. Okay, then in today's part, I will discuss about depending on the part used for culture. In that three, that is organ culture, hairy root culture, and third one is protoplast culture and somatic hybridization. Okay, these three parts comes under depending on the part used for culture. In organ culture, detailed about this organ culture. In that first part, first point in that organ culture, first, that is root tip culture, that is meristem to a meristem root tip culture. Okay, so root apical meristem is mainly responsible for the growth of roots by cell division, cell enlargement, and cellular differentiation. Tips of the lateral roots are sterilized, excised, and transferred to fresh medium. The lateral roots continue to grow and provide several roots which after 7 days are used to experimental cultures. By this culture method, it is possible to study the nutritional requirements of roots, shoot, and root growth, conditions required for the development of secondary vascular tissues, lateral root and bud formations or nodulation, etc. These are the organ culture. In that root tip culture, so root apical meristem is mainly responsible for the growth of roots by cell divisions, cell enlargement, and cellular differentiation. In that tips of the lateral roots are sterilized, excised, and transferred to fresh medium. The lateral roots continue to grow and provide several roots which after 7 days are used to experimental cultures. By this culture method, it is possible to study the nutritional requirements of roots, shoot, and root growth. Okay, and conditions required for the development of secondary vascular tissues, lateral root, and bud formation and nodulation. Then next, that is shoot tip culture in organ culture मध्ये. Shoot tip culture, that is meristem shoot tip culture. In that the shoot apex or shoot tip, that is 100 to 1000 micrometer long. In that consists of the apical meristem and one to three adjacent leaf primordia. Actively growing shoot tip is surface sterilized and is placed on a defined culture medium under sterile conditions. कोणकोणते प्लांटचे आपण पार्ट यूज करणार आहोत प्लांट टिश्यू कल्चरसाठी ते आपल्याला काय करायचे आहेत, इनलिस्ट केलेले आहेत आणि त्याचं डिस्क्रिप्शन लिहायचं आहे. ओके, सो shoot node culture is simplified form of shoot culture, which is yet another method for production from pre-existing meristem. It is the simplest method, but it's associated with the least genetic variations. Some of the crop species that have been freed of viruses by this technique, they include soybean, sweet potato, sugarcane, and rhubarb. So is this method is used with both monocot and dicot species. In that, shoot node culture is simplified form of shoot culture, which is yet another method for production from pre-existing meristem. It is the simplest method but is associated with the least genetic variation. Some of the crop species that have been freed of viruses by this technique. They include soybean, sweet potato, sugarcane, and rhubarb. This method is used with both monocot and dicot species. Then next, that is leaves or leaf primordia culture. In that, the growth of leaves on the culture medium depends upon the stage of the leaves during excision. Excision. It is observed that explants from immature young leaves grow better than explants from older leaves. So leaves 800 micrometer long may be detached from shoots, surface sterilized and placed on a solidified medium where they will remain in a healthy conditions for a long period. It is believed that leaf culture depends upon the physiological state and the age of the leaf. The shoot forming potentials differ in the leaf cultures as per the derivation of the explant and the hormonal factors involved in the plant cultures, plant tissue cultures. ओके, सो लिव्स किती प्रमाणात ते मोठं असलं पाहिजे याच्यावरती सुद्धा त्याचं जे नवीन प्लांट तयार होणार आहे ते डिपेंड असणार आहे. ओके, ते हेल्दी असलं पाहिजे आणि लॉंग पिरियडसाठी ते टिकून राहिलं पाहिजे. ओके, then next, flower culture. In that, meristem or floral cultures. In that, flowers 2 days after pollination are excised, sterilized by immersion in 5% calcium hypochlorite, repeatedly washed with sterilized water and transferred to culture tubes containing an agar medium. Flowers 2 days after pollination are excised, sterilized by immersion in कशामध्ये ठेवायचं आहे ते 5% कॅल्शियम हायपोक्लोराइड रिपीटेडली washed with sterilized water and transferred to culture tubes containing an agar medium. When cultured or such flowers produce fruits, larger fruits are obtained on medium supplemented with growth supplemented with growth hormones, and flowers excised before pollination do not produce fruits. So these culture system is useful in studying microclimates or nutritional effects on the vegetative and reproductive processes of the plant. Okay, then next, that is anther and pollens culture. So young flower buds are removed from the plant and surface sterilized. Immature stage of anther or late stage of anther containing starch filled pollen usually grow abnormally, or the development is generally ineffective, and hence for better response always select mature anther or pollen. The anthers are then carefully excised and transferred to an appropriate nutrient medium. The anthers are generally cultured on a solid agar medium where they develop into embryos or another culture under alternate light and dark period. Anther or pollen grains of different species have been successfully cultured to obtain large number of haploid plants. So pollen grains removed from the anther either mechanically or by naturally dehiscence, that anthers placed in 5 ml of liquid medium in a petri dish containing pollen grains in the culture media are sealed with parafilm and incubated. After incubation, haploid plantlets are developed. Okay, in that anther and pollens culture. म्हणजे जर आपल्याला एखादं प्लांट प्रिपेअर करायचं असेल तर त्याचं सगळे जे त्या काही प्लांट्सचे किंवा फ्लॉवर्सचे फ्रूट्सचे जे काही पार्ट्स आहेत त्याचं आपल्याला काय करता येईल टिश्यू कल्चरने आपल्याला नवीन प्लांट किंवा कल्टिवेशन्स करता येईल ओके देन next that is ovule and embryo culture in that mature embryos are excised from ripened ovule seeds and cultured mainly to avoid inhibition in the seed for germination. Very small globular embryos require a delicate balance of the hormones. Embryo is desiccated from the ovule seed and put into culture media. This type of culture is relatively easy as the embryos require a simple nutrient medium containing mineral salts, sugar, and agar for growth and development. Also, multicellular immature embryos are dissected out and cultured aseptically to obtain viable hybrids. Once the embryo is rescued, two genomes are needed to be combined together to produce a fertile plant. By this method, dormancy period of seeds can be shortened as well as haploids can be produced. By ovule culture, it is possible to grow, study various nutritional requirements and stages young embryos or zygote. So ovule and embryo culture in that mature embryos are excised from ripened ovule or seeds and cultured mainly to avoid inhibition in the seeds for germination. ओके ओव्हिल आणि ओम्ब्रिओचं सुद्धा काय केलं जातं कल्चर केलं जातं ओके देन ह्या ज्या सगळ्या टेक्निक्स आहेत हे जे काही कल्चरच्या स्टेप्स आहेत ते तुम्हाला पूर्णपणे लक्षात ठेवावे लागणार आहेत ओके देन ovaries culture in that ovaries excised after pollination can produce fruits on a simple medium containing mineral salts sugar and vitamins. Ovaries taken from unpollinated flowers fail to produce fruits on such a simple medium but can develop into seedless fruits on a medium supplemented with hormones. So by these culturing methods, physiology of fruit development can be studied. Then haploids can be produced. Then rare hybrids can also be produced by ovary culture. Dormancy period of seeds can be reduced. So these are all about the ovaries culture. Okay. Then nucleus culture. In that nucleus culture in that tissues excised from unfertilized ovules. Unfertilized ovules have no advantageous or adventive embryos if cultured on a medium containing malt extract and adenine embryoids are formed, but they are unable to germinate. When such embryoids are excised and cultured on a medium containing gibberellin, plantlets may be formed which can then be transplanted to the field. So disease-free clones can be obtained by nucleus culture. ओके त्याच्यामध्ये आपण ग्रोथ हार्मोन्सची सुद्धा त्यांना ॲड करू शकतो आणि नवीन प्लॅन्टलेट बनवू शकतो. ओके. देन सीड कल्चर, in that the seeds are treated with 70% alcohol for about 2 minutes. The seeds are treated with 70% alcohol for about 2 minutes, washed with sterile distilled water, treated with surface sterilizing agent for specific period, once again rinsed with sterilized distilled water and kept for germination by placing them on double layers of pre-sterilized filter paper. So placed in petri dish, moistened with sterilized distilled water or placed on moistened cotton swab in petri dish. So the seeds are germinated in dark at 25 to 28 degree Celsius, and small part of the seedlings is utilized for the initiation of callus. The seeds are treated with 70% alcohol for about 2 minutes, washed with sterile distilled water and again treated with surface sterilizing agent for specific period of time. Then once again rinsed with sterilized distilled water and kept for germination by placing them on double layers of pre-sterilized filter paper placed in petri dish, moistened with sterilized distilled water or placed on moistened cotton swab in petri dish. The seeds are germinated in dark at 25 to 28 degrees Celsius, and small part of the seedling is utilized for the initiation of callus. Then next one is cotyledon culture. In that immature cotyledon develops into somatic embryos, shoot buds and complete plants if cultured on a suitable nutrient medium. These are the 10th point in organ culture. Okay. Then endosperm culture. In that if endosperm is cultured on a proper nutrient medium, continued proliferation of callus mass takes place and subsequently plant is regenerated. It is useful for plant breeding and horticultures. It also useful in the production of plantlets as an alternative to the conventional methods of crossing, that is tetraploids with diploids or triploids induction, which is applicable to fruit trees. Okay, these are the endosperm culture. Okay, then fruit culture. In that the culture of fruit tissues as whole organ or isolated tissue sections such as ovary has been successfully cultured to give rise to mature fruits. Example, that is strawberry. Usually when an isolated portion of the fruit tissue is introduced into a sterile environment, it immediately loses structural integrity and degenerates into a rapidly dividing callus mass. Loss of structural integrity is associated correspondingly with an alteration of physiology that is subsequently reflected in the production of an altered metabolism. So it is not possible to make a meaningful study of fruits developing using callus derived culture. So the use of fruit culture is to serve as bioassay system to study fruits maturation events within a controlled environment. So these are all about the fruit cultures. Okay. Then next one, that is plant cell culture. In that culture of isolated cells or very small aggregates remaining dispersed in liquid medium. Okay. These are all about the organ cultures in types of tissue cultures. Okay. These are the all points included in the organ cultures. ओके. सो ऑर्गन कल्चर्समध्ये आपण 13 कल्चर्स वेगवेगळे बघितलेले आहेत. ओके. सो डिपेंडिंग ऑन द पार्ट यूज्ड फॉर कल्चर, हे जे 2 पार्ट आहेत ते आपण नेक्स्ट लेक्चरमध्ये बघणार आहोत. सो थँक्यू फॉर for fully watching this video. And if you have not subscribed to my channel, then subscribe first and press the bell icon to get latest updates of upcoming videos. So thank you. Happy learning. Stay safe. Thank you so much.